High cholesterol levels are a global concern, contributing to about 2.6 million deaths annually, as reported by the World Health Organization. In Canada, 2 in 5 adults suffer from unhealthy LDL cholesterol levels, which can lead to plaque buildup in arteries and increase the risk of heart diseases and strokes. While medication such as Praluent or statins can help address cholesterol concerns, they are costly and have adverse side effects such as muscle pain. That is why our multidisciplinary team from iGEM Calgary is developing a novel solution, CholesterLock, a modified protein that will lower LDL cholesterol levels by blocking the NPC1L1 protein receptor. By combining synthetic biology with engineering techniques, we aim to mitigate the problem of high cholesterol and help millions live healthier, longer lives.
When it comes to managing high cholesterol, a healthy diet and exercise are the first line of defense. In an interview with family clinician Dr. Lowen, he stated that lifestyle modifications can be effective for many patients, but for those who cannot manage high cholesterol with lifestyle changes alone, medication may be necessary. However, for one patient we spoke with, statins caused extreme side effects. The patient stated that their experience with statins was causing them extreme joint pain, and they felt like they had aged 30 years. The impact on their quality of life was so severe that they made the difficult decision to discontinue the medication. The patient even expressed a preference for the increased risk of a heart attack over continuing with the debilitating side effects. While this patient's experience is unique, it highlights the importance of understanding that every patient responds differently to medication. After the patient experienced extreme symptoms while taking statins, they consulted their doctor for help. Their doctor referred them to a cardiologist who prescribed them an alternative medication called Praluent. While it was effective in lowering their cholesterol levels, it came with a high price tag, $750 a month for the injection. It's clear that there is a need for an alternative cholesterol medication that can effectively lower cholesterol levels without causing adverse side effects.
Our solution is an Fc fusion protein that blocks NPC1L1, a receptor responsible for the absorption of cholesterol in the lining of the small intestines, and indirectly involved in saturated fatty acid absorption. Our engineered protein CholesterLock involves a cholesterol connected to the cholesterol isoprostane domain of the hedgehog protein that is linked to the Fc region of immunoglobulin G. The purpose of using hedgehog protein is to attach a cholesterol molecule that binds to and plugs the opening of NPC1L1 to prevent the continued uptake of cholesterol. The significance of immunoglobulin G is to act as a transporter protein, with studies having shown a high stability in gastrointestinal conditions. Overall, this process forms CholesterLock, a newly engineered Fc fusion protein which blocks cholesterol uptake through a lock and key mechanism. Our solution focuses on the UN Sustainable Development Goals, with a primary focus on goal number 3 of good health and well-being. By reducing cholesterol absorption, we aim to promote good health and well-being for millions of people affected by high cholesterol levels. We also aim to tackle goal number 10 of reducing inequalities and goal number 12 of responsible consumption and production, through a bacterial-based process requiring fewer resources, leading to a more cost-effective process for large-scale production.
Initially, we had planned on delivering CholesterLock through probiotic bacteria. However, after consultations with a biomedical engineering professor at the University of Calgary, we decided to shift towards a purified protein pill delivery system. Firstly, the harsh stomach conditions pose a challenge to the bacteria's survival. Therefore, we will design a protein pill designed for the pH of the digestive tract by incorporating an existing protein that is resistant to denaturation under gastrointestinal conditions. It would also have been very difficult to facilitate post-translational modifications in the bacteria in the digestive system, coupled with the fact that bacteria don't produce their own cholesterol for the modification. Secondly, by producing the protein outside of the lab, additional modifications such as affinity tags can be added to help with the purification of the protein. Thirdly, because there are other types of bacteria in the digestive tract, we would have needed to consider the interactions that our bacteria would have had with them, as well as if it might compete with them. However, by working with a purified protein, we mitigate some of these biosafety concerns. Overall, expressing just the protein will simplify the delivery process, avoid biosafety concerns, and increase our protein purity and yield.
Instead of completely computationally modeling our potential protein around the unknown factors such as pH, body temperature, stability, and function, we decided to explore previous research done on therapeutic protein delivery to find a chaperone protein candidate. In this research, we found that the fragment crystallizable Fc region of immunoglobulin gamma 1, or IgG, was a widely used domain for a chaperone in what is called an Fc fusion protein. This is because the domain has high stability in the gastrointestinal system's low pH and high temperatures. Here you can see the 3D model of IgG, and Fc fusion proteins are most often produced in Chinese hamster ovary cell cultures because of these sugar residues in the lower Fc region which are added post-translationally. These modifications are very difficult to replicate in bacteria and pose a challenge to the development of CholesterLock. But in the research on IgG, we found that they are only required for IgG's original function as an antibody, and not including them has little effect on stability. This means that we are able to produce the protein in bacteria, saving costs and hassle associated with using cell cultures. After finding a suitable chaperone to accompany our cholesterol-modified region, we needed to evaluate the cholesterol modification process. Luckily, the literature shows that the C-terminal end of hedgehog proteins, which we will be using, make the process relatively easy by self-cleaving in the presence of cholesterol or similar molecules. The only problem is that the side of the Fc region that linked proteins are usually added was opposite to what we needed to leave the cholesterol exposed. Once again, the benefits of working with a protein widely used for this exact purpose is the extensive amount of research that has already been done to overcome these challenges. We found a study demonstrating that attaching proteins to the opposite end of the Fc region has almost identical expression rates as the original and retains stability at body temperature for over 70 days. The effects of freeze-drying the protein were also investigated, showing no loss of function, which is an important factor for implementation in pill form. After connecting the proteins with a placeholder linker sequence, we used modeling software to provide insight on the 3D structure of our first prototype protein. As the Fc region of IgG is homodimeric, meaning it binds to an identical copy of itself, we used a separate modeling software to visually represent what the protein might look like before cholesterol modification. Confirming the stability of the protein will now mainly be done in wet lab experiments, and regarding our protein's function, we plan on using membrane scaffold proteins to make structures known as nanodiscs, which are capable of solubilizing the otherwise insoluble NPC-1L1 in solution while leaving both ends of the protein accessible for testing. To analyze the binding of CholesterLock to NPC-1L1 and monitor activation or inhibition, we plan to use linked fluorescent proteins on CholesterLock, NPC-1L1, and a protein that binds to the sequence on NPC-1L1 that is exposed when activated, to monitor the interaction of those proteins using fluorescence confocal microscopy.
The next short-term goals for CholesterLock include further development of our prototype. We will begin by producing NPC-1L1 receptor proteins in a cell-free system or ordering the receptors themselves. After that, we'll be investigating the nanodiscs, as in figuring out their exact specifications such as diameter, and creating them custom-built for the structure of NPC-1L1. This will allow us to suspend the receptor and conduct experiments with it. The next milestone for CholesterLock success is finding the right sequence for the linker between the antibody fragment and the cholesterol modifying domain. This is an essential component of the CholesterLock fusion protein and needs to be functional at the pH of the digestive tract, it needs to stay linear, and it needs to fit into NPC-1L1 without interfering with the function of the cholesterol modifying domain or the Fc fragment. Altogether, these goals are stepping stones to assembling CholesterLock and testing its interactions with NPC-1L1. In the long term, if this experimentation reveals that our current design doesn't interact well or work as predicted, we plan to pivot to an alternative form of competitive inhibition, or instead, to a proteolytic mechanism to inhibit NPC-1L1.